In this video, we're going to look again at the central dogma, but this time we're going to concentrate on the flow of information from DNA to RNA to protein. And we're going to look at the actual information, in other words, the sequences of nucleotides in DNA and RNA, and the sequence of amino acids within a protein. Okay, so let's start with the double-stranded molecule of DNA. And we have some sequence data here. This is just a tiny part, let's say, of a gene. So you can see we have a double helix on either side. And in the middle, that double helix has been pulled apart. We can sort of think of this as maybe a transcription bubble. And we're going to get ready to transcribe a molecule of messenger RNA. Okay, so we have two strands of DNA. And there's two things I want you to notice really quickly here. Number one is that our strands are running anti-parallel to one another. So the top strand is running from 5' to 3', and the complementary strand is running 3' to 5'. Secondly, we're using our base pairing rules. So if you look at the sequence, you'll see that A and T are paired. So if we have an A in the top strand, then we'll have a T in the bottom strand. If we have a T in the top strand, then that will pair with an A. G pairs with C, and C pairs with G. Okay, let's think about how the sequence, how this information is going to be copied onto a molecule of messenger RNA. And to do that, first we need to know which strand is actually going to be used by RNA polymerase II, if we're a eukaryote here, in order to make the messenger RNA. Now, you're not going to know which strand is which simply by looking at it. This is an extra piece of information that you're going to have in order to predict the sequence of messenger RNA. Okay, so we are going to label the strands for you. The top strand is going to be the non-template strand, and the bottom strand will be the template strand. What do those two things mean? Well, that strand on the bottom, the template strand, it's the template because it is what RNA polymerase is actually going to use, physically interact with in order to determine the sequence of messenger RNA. By the way, there are different names for these strands, and your instructor might be using one of them. Template strand can also sometimes be called the non-coding strand or the antisense strand, and there's other names as well. In contrast, the strand on the top is called the non-template strand. This is also sometimes called the coding strand or the gene strand or the sense strand. There's other names as well. I'm sorry about that. I'm just labeling them template and non-template to be as clear as possible. And this strand is not going to physically participate with RNA polymerase while it transcribes. All right, so let's start transcribing. And like I said, RNA polymerase is going to use that bottom strand. What it's going to do is apply the complementary base pairing rules that you've already learned. Okay, so our first few bases are T, A, C, G, meaning that RNA polymerase is going to see the T, and it will add the complementary base. So that would be A. For A, it would add U, because recall that RNA has uracil rather than thymine in it. For C, it would add G, and for G, it would add C. All right, so this is in fact what the sequence would be. Now, there's actually an easier way to predict the sequence of messenger RNA, and maybe you see it now that these sequences are up. The easier way to predict the correct sequence of messenger RNA is simply to look at the non-template strand on the top and copy it down exactly, with the only exception being replacing every T with a U. So notice how these two sequences are otherwise identical. Notice also they have the same polarity, okay? So if RNA polymerase is using the template strand, then it's going to be making an anti-parallel strand that applies when we have a DNA-RNA hybrid, as well as when we have double-stranded DNA. And so the result is going to be this messenger RNA sequence that is a faithful replica of the non-template strand, both in terms of polarity and in sequence. Okay, let's move on now to translating this strand into a polypeptide. Remember, a polypeptide is a string of amino acids that can be folded up to form a functional protein or perhaps a subunit of a functional protein. All right, and to do that, we're actually going to think of this sequence in chunks of 3. Every 3 nucleotides is what we call a codon. Okay, so every 3 nucleotides is going to code for a single amino acid in this step of translation. Okay, so to understand which amino acid will be used for each codon, we're going to consult a genetic code table. Because there are 64 codons, this is certainly something that you would never need to memorize. Genetic codes are widely available. You would simply pull one up and use that. Okay, so let's start with the first codon, which is AUG. This one we've already filled in for you. And to read the genetic code, we simply look at the first letter first. That's going to be in the rows of the table. The second letters are in the columns, okay? And that's going to dial us into one of the 16 boxes for a total of 64 codons in the genetic code. So AUG, we can see is in the third row because the first letter is A. It's in the first column because the second letter is U. And in fact, this one's easy to see because it's highlighted in red. This is going to code for methionine. The 3-letter abbreviation is MET, and the 1-letter abbreviation is M. All right, this was highlighted in red because methionine is going to be the first amino acid added to a polypeptide, always. Now, sometimes it will later be cleaved off in post-translational processing, but at least to start with, all polypeptides are going to start with methionine. All right, let's go to the second codon, which is CCU. Go ahead and try to find that on the codon table. Okay, so first letter C, that takes us to the second row. Second letter C takes us to the second column. And CCU, all 4 of the codons in this box, in fact, code for proline. Next codon is UAC. And hopefully you came up with tyrosine. Next codon, AGC. So we're down to the third row. We're all the way to the 4th column. AGC codes for serine. UGC. All right, so first row, last column. It's going to code for cysteine, UCU, serine, and CUG, leucine. Notice also in the first row how there are 3 stop codons. Any of these is going to signal termination of translation. So in this case, the ribosome would simply stop translating. Okay, so that is it for now. Hopefully you've learned how to predict the sequence of messenger RNA as well as the sequence of a polypeptide.